Okay, so today the topic that we are going to study is on host pathogen interactions. We have already learned about the different microorganisms that are associated as normal flora with our body or with the host. Now, today we will see how the interaction between a pathogen and a host takes place. So let us go ahead with that. Now, microbes are found everywhere. On a daily basis, we ingest, inhale, and transport thousands of organisms. Those organisms may be bacteria, virus, protozoa, or parasites. Most of them have no ill effects due to the protective mechanism that we have in our body. Mechanisms such as coughing, urinating, sneezing, and defecating. And humans and animals, they have friendly organisms throughout their bodies that survive as normal flora, and they colonize a host but do not cause any disease. However, if these organisms begin to cause any obvious damage to a host by invading tissues or producing toxins, then they become pathogens. So this is what the difference between a pathogen and a normal flora is. A normal flora, when it's in a body, it will not cause any harmful effect on our body because that is its home. And also we have our own immune system that will help to fight against any kind of infection that may arise in our body. And those normal flora also acts as a protective layer or protective organism who helps in fighting against the pathogens. Okay, and so that is why this normal flora are not considered to cause any kind of damage until and unless the organisms they start invading the tissues, and then they start producing toxins and they become pathogens. Now, pathogens, whichever organism is a pathogen or will intend to cause any infection in your body, those kind of organisms will always try to invade the tissue. They will try to break your tissue, or they will try to enter through any opening in a tissue and start causing infection in your body. They will also produce some toxins, and those toxins are hazardous to our body that will lead to certain disease or infection. Such kind of organisms are called as pathogens. What is the significance of the host pathogen interaction? Host pathogen interaction, it provides information that can help scientists and researchers understand disease pathogenesis, the biology of one or many pathogens, as well as the biology of the host. It is through this interaction that basic research discoveries are made. That means, in order to learn about a particular pathogen, or in order to find out how that pathogen can be killed, or if you want to know about all the information about a pathogen, then you will have to understand the interaction that takes place between the host and the pathogen. How does the pathogen enter the body? Every pathogen will have different kind of routes of entry. Every pathogen will have different kinds of tendency to infect a person. So that is the interaction that you must understand, or that information must be known. That will help a scientist or a researcher understand the disease pathogenesis. Pathogenesis means the medium how an infection can happen. Okay, for example, a respiratory tract infection causing bacteria. That bacteria will always enter your body through the respiratory tract, through your nose. Most of the time it's through the nose, and sometimes it may be through the mouth. It will not get into your body along with your food. And even if it enters to the food, then it will not be able to cause that respiratory tract infection, because it will reach your intestine, and that tract of the intestine, the tract of the respiratory organs, those are completely different. right? So that cannot cause any harm in the intestine because its target organs are the respiratory organs. So that is how that is how you should understand its pathogenesis, and then only you will be able to learn more about diagnosing the infection, or how what kind of antibiotics may be used to treat that infection and so on. So the interaction part is very important. Now, moving ahead with the Koch postulate. Now, you all must have learned about Koch postulate even before. So this is an experimental step that was used by Koch to determine disease causation. Okay. Now, disease causation. See, there are various diseases that are caused by different infection, by different microorganisms. But how do we determine that a particular microbe is the specific one for a particular disease? Every microbe is specific for a particular infection. So you must be able to identify that. Once you identify that, that will help you to diagnose the infection more properly, diagnose the organism more properly, and also that will help in treatment of the person or treatment of the patient. So that is why this Koch postulate is a very important step that helps us in determining the exact microorganisms responsible for causing any infection. So the first point in Koch postulate was the microbe must be present in every case of the disease. If a particular disease is caused to a person, or if a person is infected by a particular microorganism, then the symptom that it shows, at every step of that symptom, the microorganism must be present in that host. Or if the same symptoms is being shown in some other person also, the same kind of microbes must be present in the host. That is the first case. If that first factor is satisfied, then at one point we can say that yes, that particular microorganism is responsible for causing this infection. Okay, but again, only depending upon one point we cannot decide this. Okay, so we will also again have to fulfill the second point. The organism must be grown in a pure culture from the diseased host. And once you isolate that microorganism from the person who is diseased, then that microorganism must be grown in a pure culture. Okay, we have done that. Then after doing the second point, we move towards the third point. The third point is that the same disease must be produced when a pure culture, the organism, is introduced into a susceptible host. So if you are now inoculating that organism, that pure culture which you had grown on a media, now you take the pure culture and then inoculate that culture into a host, into another host. Okay, now when you inoculate that culture into that host, then the disease that is caused because of the inoculation of that particular organism must be same that you had seen in the very beginning. It's the microbe, when you were isolating from a diseased person, you have seen some symptoms, you have seen the disease. So that similar disease must cause to that susceptible host into whom the organism is being inoculated. Then the fourth point is the organism must be recovered from the experimentally infected host. And again, the same organism must be able to be recovered from the person or from the host that was experimentally infected. So if these 4 points are fulfilled, then we can determine that a particular organism is responsible for a particular disease. Okay. So this kind of experiment is done when basically it is done when you identify a very noble organism, or you are identifying a new organism. Like now in this present scenario, if you're talking about COVID, yes, coronavirus was prevalent from many years It's not a new virus, but the strain COVID-19 was a new one. So probably, I don't know how they did it, but the researchers or scientists, if they are to determine whether the ongoing pandemic was because of COVID-19, so probably at the very beginning, they also must have done similar kind of experiment in order to determine that the present pandemic is because of that COVID-19. So once that was determined, then no, it was then published or it was released worldwide that yes, COVID-19 is the causative agent of this particular pandemic that's going on. So this is how you determine the causation of a disease, or you determine the microbes who are responsible behind the causing of a particular disease based on the Koch postulate. So now when we perform or if you want to determine such kind of experiment, we cannot perform it on a human being. So that is why it's always that experimental animals that are taken, and here in this case they have taken a mice. So this was a mice which was already diseased. Okay, it was showing some kind of symptoms. So they wanted to identify the organism which was responsible for causing the disease in this mice, and then the the organism was isolated from this, and then it was grown. A pure culture was grown on using this media. Then when the colonies were shown, those colonies were again injected into this susceptible host, and then this host, it showed similar symptoms as that of the mice from where the bacteria was isolated. And then from this mice again, the bacteria or some bacterial sample was isolated, and again it was grown on a pure as a pure culture, and then it shows the similar kind of growth which was seen on the previous clip. So this helped to determine that yes, that particular bacteria was the one which was responsible for causing the infection in this mice. In all of them, actually, it was the same bacteria related with the same disease. Now, the Koch postulate is a good is a good way how you can determine the organisms responsible for a disease, but then again there are some exceptions to this criteria. Now those exceptions are first of all, some organisms cannot be cultured in a lab and grown on artificial media, so that causes a problem if that particular organism cannot be cultured in a lab. Then some pathogens, they cause several diseases. Now the conditions they show are different kinds of conditions. So it becomes very confusing to identify again. Some pathogens, they cause several disease conditions such as Mycobacterium tuberculosis, which can cause lung disease and other disease of the skin, bone, and internal organs. Had it been only related to the lung, then no, that was not a major problem. But then again, it causes diseases in the skin also, bone also, internal organs also. So that immediately does not help us identify the bacteria. There may be ethical reasons that do not allow testing, that is, human diseases with no animal model, like for example smallpox and rubella. This kind of viral infection, it does not show any symptoms in animals, so how are you going to find it out? So that is what some exceptions are. That is why when I told about COVID, I said that I probably don't know whether they followed the same postulate or not, because COVID is seen in human beings, so how are they going to find that out? They will not be able to do that on animals. So they must have found out some other way to determine that the organism and the disease. Okay. So now if you want to study about the host and pathogen interaction, there some points are kept to be mind. The points that are required to be kept in mind are invasion of the host through primary barriers, how the host is invaded, whether the primary barriers are destroyed in order for the organisms to enter or invade a host. Second, evasion of local and tissue host defenses by microorganisms. Then thirdly, microbe replication with or without spread in the body. How are the microbes replicating? Whether it is replicating by spreading throughout the whole body, or it's just replicating within a particular organ or particular cell. A host immunologic ability to eliminate or control the microbe. But how strong is that person? How strong is the host? Whether his immune system is strong enough to eliminate or control the microbes. So all of these components are responsible for the host and pathogen interaction. And individuals or animal skin and mucous membranes generate an environment for microorganisms to interact with the body. It is, see, we have learned in immunology that skin and mucous membrane, they are a part of the barrier, physical barrier of our body, and so it plays a major role in order to prevent many infections, and also they are the one who interact with the microorganisms in the first place. So this interaction between the host and the organism is referred to as symbiosis. This is called as symbiosis. See the way how your normal microflora or any other organism, they happen to stay on the skin. They happen to reside in the skin. So it is giving a platform for this organism to live there on the body itself without getting harmed. So this is a relation that the host and the organism both have built amongst themselves. So that particular interaction is called symbiosis. So now the symbiosis or symbiotic relationship can occur in three different forms that can occur at anatomical level. In order for this to take place, the following components will need to occur. So what are the three different forms on which the symbiosis is based upon? First one is mutualism. In mutualism, what happens? Both the microorganism and the body, they both work together. For example, relationship would be cows and the bacteria in their rumen. Bacterial cellulose facilitates digestion in the animal, while the bacteria benefit from nutrients in the rumen. So they are both dependent upon each other. Nobody is harming the other one. They are both dependent and both helping each other. So this is a mutual understanding between the bacteria and the host. Okay, so that kind of relationship or that kind of symbiotic relationship is called mutualism. So here what is happening? The bacteria which is present in the cow, okay, in the intestine of the cow, those bacteria, they are helping the cows in the digestion. The cellulose which is present in the bacterial cellulose, it's helping in digestion in the animal, while the bacteria itself is also getting benefited. How? They are getting the nutrients from the rumen of the cow. So that is called as mutualism. Next is commensalism. What is commensalism? In commensalism, either the body or the microorganism benefits, while the other is not affected by interaction. That means any one of the two is getting the benefit, not both. So this is not mutualism. It's just commensalism where both are living together, but then only one is getting the benefit, while the other is neither benefited nor harmed. Okay, so that is called commensalism. For example, this includes microorganisms that make up the normal flora that inhabit the eyes. These organisms, they live on the secretion and dead cells, but they do not affect the host. Okay, so there are some microorganisms that may be present in our eyes. Those microorganisms does not cause any infection to the eye. They just live upon the secretion and the dead cells that are present nearby to the eyes, but they are not causing any harm to the host, nor the host is causing any harm to those bacteria. Okay, so that is called as commensalism. Thirdly is parasitism. In parasitism, one organism benefits at the expense of the other. When you say at the expense, that means the other one is getting harmed for it. For example, parasites use the gastrointestinal tract of a human or animal as an environment in which to reproduce. Okay, so here the parasites, they include human or animals' gastrointestinal tract in order to complete their life cycle. And in order to complete their life cycle, they will have to harm a human, or they have to harm an animal. Okay, so in this process, the one organism which is getting harmed here is the host. Okay, so but they are getting no benefit. All the benefit is taken up only by the parasite. So this kind of relationship is called as parasitism. So these are the three different symbiotic relationships that can occur at an anatomical level. Okay. Then yes, the body's normal flora. We have already studied about the body's normal flora. That there are two types of normal flora that reside. One is the resident flora. Then another one is the transient flora. The resident flora, they will survive for extended period. Transient flora, they are temporary. They do not reside for a longer period of time. Normal flora help to provide defenses against invading pathogens by covering adherent sites. They producing compounds toxic to other organisms and preventing pathogens from consuming available nutrients. So that is the role of the normal flora. It's protecting, as I've been telling you all. For disease to occur, there must be a change in the body's environment, which in turn allows the pathogen to overcome the normal flora. This can occur through a change in the pH of the body or elimination of normal flora due to antibiotics. Our body is meant in a way so that the normal flora can survive on our body. So the same temperature that we have for us, the same temperature is required for normal flora. The same pH that we have in our body, the same pH is required for normal flora, and so on. So if we are to get any infection or disease, there has to be some changes in our body. Maybe in the change of temperature, maybe in the change of the pH, or any kind of wound or something that will help or give an entrance for the pathogen to enter into our body. So that is how we get infected or disease. And then obviously the normal flora is always there to fight against those pathogens. But then sometimes they cannot. Why they cannot? Because they become weak when our body's conditions changes. And also sometimes we are upon antibiotics and all. So those antibiotics also have an adverse effect on this normal flora. And too much consumption of antibiotics may destroy the normal flora in your body, and then that will again lead to exposure to different kind of pathogenic bacteria. Now the host defense. A microorganism will not be able to invade unless it overcomes an animal's or individual's host defense. Host defense means the defense mechanism that the person has, or it may be in the case of animals. So animals also have their own host defense. So until and unless our host defense becomes weak, a microorganism will not be able to invade us. So the host defenses in our body may include skin and mucosal secretion, non-specific local responses, non-specific inflammatory responses, specific immune responses, and these are the host defenses. The ability for a pathogen to overcome host defenses can be accomplished by two distinct components. A primary pathogen that may cause disease in a healthy host, or opportunistic pathogen which cause disease if host is immunocompromised. That means what? How will you know that a particular pathogen is entering inside your body because your host defense mechanism has become weak? This can be found out by two ways. It depends upon the organism which is causing the infection or causing the disease. If the infection is caused in a healthy body, then the infection or the bacteria is going to be a primary pathogen. Primary pathogen means something that is entering from outside and then it fought against your immune system, but then ultimately at one point of time your immune system got defeated by those pathogens. Okay, so that does not mean that you have a very weak immune system. No, your immune system was present, but always the immune system cannot win. Okay, sometimes it gets defeated by the pathogens. So if that is the case, then the pathogen that is harming you is a primary pathogen. But sometimes what happens, there are certain opportunistic pathogen that enters. Opportunistic pathogens means what? They are normally not pathogens, but then if they get an opportunity, then they may turn into a pathogen. And this most of the time happens with hosts who are immunocompromised. Immunocompromised means those whose immunity has become very weak. For example, a person suffering from HIV, that person's immunity is already weak. Okay, if that person's immunity is already weak, so even the even the bacteria, those which are residing on our body as normal bacteria, they may also turn to cause an infection, and then those kind of bacteria will be termed as opportunistic pathogen, because the host immunity became weak, and then the bacteria, it is taking that opportunity to cause infection in the host. Okay, so this is about the host defense. Then pathogens defense. What is pathogen defense? Pathogen, it contains virulence factors that promote disease formation and provide the opportunity for a microbe to infect and cause disease. The greater the virulence, the more likely diseases will occur. Virulence factor